This is the third of our inaugural lecture series in the School of Engineering, and this is an initiative that has led by our Director for Research and Innovation, Magda Jukovic. She conceived of this idea. I think it's a fantastic initiative that the school runs, and the whole idea behind this is to celebrate when people are promoted to the grade of professor. And in particular, it's an opportunity to reflect on their research achievements, because a large part of that promotion is based on research excellence and research achievement. So it gives us a lovely opportunity to come together and celebrate the promotions, to allow others in the school to hear the research achievements of these individuals who have made it to this grade. So, you know, we're a busy school, we're all busy teaching, and we're all in one building, but actually, when you start to think about it and talk to people, we don't get enough exposure to the types of research activity that people are doing. Now this is moved on. So I'm going to get started then. So today, we have two lectures, inaugural lectures today. So Professor Guangxue Wu has agreed to tell us about Microbial Niche Nexus, the Art and Science Environmental Biotechnology. So Guangxue has a PhD from Tsinghua University, and a PhD from University of Galway, and a master's degree from Tsinghua. He also was an assistant professor in Tsinghua and guest associate professor at Kyoto University. His research focuses on sustainable wastewater treatment and sustainable development, and he convened ISO and received the ISO Excellence Award in 2020. I believe he's probably joined by his wife and family here today, so that's great to see you guys. So welcome. So maybe it'll be nice to hear from Guangxue about his own achievements as well. So I'll hand it over to you now, and is his presentation set up here for him to go?
Yeah.
Okay.
Thank you very much, Lisa, for the introduction, and also for Magda, for organizing, this lecture. So. Good evening, everyone. My name is Guangxue Wu. So, I think it's truly my honor, today to be here to talk, my research. So, the title of my talk is Microbial Niche Nexus: The Art and Science of Environmental Biotechnology. So, this idea has been, formed for a long time. I think just throughout my academic career, the deeply, the understanding of this is just when we ask Microbial, to clean water and also to recover resources from wastewater, or maybe to generate, that renewable energy. So we are not simply that copying nature. We need to shape, that environmental ecosystems. So this is the basic idea. Something like we need to introduce more types of that microorganisms in that system, and also we need to make sure that more functions can be performed in that engineered environment. To achieve this, I think at least two aspects. One is that the art or engineering, we need to design the system that to make sure that microorganisms can survive and also that perform their function. And at the same time, we need to understand, to apply that science to understand, how these microorganisms, what's their behaviors and what's their interactions, and also it's very important, to understand that ecological strategies. So by this means, we can regulate or optimize, this system, effectively. So anyway, this intersection among this art and science, when design, meets understanding, when engineering meets that ecology, that forms the core of my academic journey. So following, I will introduce 4 sections about my, background and also my contribution. This section is very important. I will introduce how I transformed from that an environmental engineer to that an environmental biotechnologist. And also, I will introduce that my ongoing activities, and also maybe just one slide for that future vision. So first, about myself, this slide shows, the 25 years of my academic career. So I was trained in water and wastewater engineering in Shandong Jianzhi University. Today, I have two classmates. They are visiting professors. And also I studied in Tsinghua University for my master, and then here, for my PhD. So my PhD supervisor was Dr. Michael Rogers. So during that time, my PhD was not that going so well because I changed the research topic halfway through. So after 1.5 years, I just changed from that drink water to wastewater. So at that time, the loss of that experience, especially I learned lots of that techniques and also that knowledge from other disciplines. I don't know, maybe some of you that don't know that NCBS, this center is very, very good because they have lots of that advanced that equipment. So at that time, I learned lots of this that microscopy or maybe others. For example, I took photos for the biofilm, from the Colorado River. So this seems very beautiful. Of course, I also learned microbiology knowledge from that Dr. Kevin Collins and also that Vincent and also other members. It's very good. At that time we have several that colleagues from Tsinghua University that including that Xinmin, Li Wen, and also Jianping, Su Huang, and also I have received lots of that help and support, including that Fred. Yeah. This is my son. At that time, he was only 2.5 years old. So now he's studying at Trinity. So the time is flying, and I just checked because the time was the best or maybe was the worst. It's regarding to which aspect. And also after that, I moved back to China, and then worked in Shenzhen, in Tsinghua University. I think at that stage, it's very important, or maybe I was so lucky to receive, lots of that mentor from, that senior, that colleagues. One is that Professor Hong Ying Hu. I think it's sometimes, it's just so lucky because I moved there in May and he moved there in July. So we have, so many years of that overlap. So I learned a lot, from him, including, we just contribute to that major project. This one is really very large scale, that around, 11 million euro. So something like that. That should be quite helpful. And also we organized, national and also that international conference, and also that, for developing that standards, national and also ISO. Another one is just, Professor Tanaka from Kyoto University, because I learned also a lot from him. We met, since 2010, but we, worked together for different, activities, since that time. And especially, we contributed to that, double master degree in environmental engineering between Tsinghua University and Kyoto University. That's I think at the beginning, even 2006 or at that time, two universities, planned this activity, but finally it's only after maybe more than 10 years. So we got this done. And also, when I worked in Shenzhen, that Professor Li Zhenxing, she's my final year project supervisor. When I worked in Shenzhen, she also helped me a lot, especially in China, because we don't have so many students. So there are more than 10 that master students, from her group joined my project in Shenzhen. So that helped me, significantly. And also for myself, I visited lots of that wastewater treatment plant. I think this front line, the visit, that's very important, for us. And after that, then in 2020, in that December, I moved back, to Galway. I think here I just want to mention once that, during that period, especially during that, application, apply this, position, I prepared this framework for my research. This is still the foundation, for my current and maybe in the following, years, that should be the structure for my research, focus. And also this slide also showed during that, interview. So now after 5 years, seems, I have achieved all these, activities, including that research, also team, members, and also that, different conference or workshops organization. I think in the following stage, hopefully we can just, get more maybe activities, and also maybe can achieve our long-term that research goals. But anyway, for this, stage, it's very important for me to know, what's my deeply, that interested that research topic, and what I should be focused on. This slide just, confirm this one. So that's my background, that's 3 stages. And then the next, I will introduce briefly about my academic contribution, and that including the challenge and also that solutions from the engineering or from that science. And then I will use that 2 examples to introduce that technology achievement and also that knowledge generation. So anyway, nowadays we are facing that different, that environmental challenges. So this should be, a problem that we need to solve. And especially, for this one, we can see, for lots of aspects, for example, for nitrogen and the phosphorus, this all have that, beyond the something like, that capacity of that our Earth can support. So that's why we have to solve the problem, including that, water pollution. And at the same time, we need to achieve this sustainability, including different aspects. But for all of this, the water, that should be the something like a bloodstream for all these, aspects. So that's why, next I will introduce about this water. For this water, we need to consider this one from at least 2 scales. One is that, large scale from that system. This is the conventional idea or concept about that water cycle. Usually, we just take water and then use water, produce this drink water. After using that, produce that wastewater, and then we treat wastewater. Finally, that discharge to that receiving water bodies. So this is the conventional, that linear, that system. But now we just try to understand how to make sure this is more sustainable. So that's why I introduced that circular, that system. Now we can regard, this wastewater as that refining rather than treatment. So we can use that refining technology or biotechnology to recover energy or maybe that nutrients from that wastewater. So, and also we can introduce, for example, that wetland system. This can make sure that treated wastewater can be changed to that natural water. This can be reused, for drinking water better than, directly from this treated wastewater. And also from that maybe microscopic, level. So we need to understand that wastewater or water from at least, these 3 aspects. One is that, quantity, usually this should be the flow rate. And also we need to consider quality of water, what's included in that water, the chemical compounds, or maybe others. And also we need to consider about this dynamic, because all these, compounds or quantity is not constant. It's changing all the time. And especially in Ireland, even we don't, lack of water that resources, but still sometimes, for example, in the summer, maybe some areas, still maybe the water resource is limited. And also at the same time, sometimes maybe that industry, that wastewater discharge can cause pollution of that river. So anyway, this just, from these two skills, we need to understand that water. Regarding to this wastewater treatment, because the technology of that activity sludge has been developed for more than 100 years. Especially during that 100 years, during that period, researchers from all over the world, just thinking about what's the future of this wastewater treatment. So that's why we summarized these three stages. For example, initially we just try to remove that pollutants from wastewater, but now we need to recover that resource from wastewater. That includes that water itself, and also we can recover that energy from these organic matters, and also we can recover that nitrogen, phosphorus from that inorganic matters. And also at the same time, it's very important nowadays, we need to think not only water, but also how to connect this water to energy, to food, or maybe to that ecology, or maybe that biodiversity. So all of this, the basis, the basic solution to solve all of this, usually that biological process, that should be the main solution, because this one costs less than chemical or physical solutions. And the basic that should be based on that microbial principles. So that's why we need to connect that microbial, that ecology, that theory or principles to that engineering. So based on this challenge, for myself, I just introduced two. One is that engineering solution for my research that focus on the life cycle of this water treatment processes, that including secondary and also that tertiary or maybe advanced. This is the main stream, and also we have that side stream. For this one, we worked on this animals, that technology, and also nowadays I'm focusing on this anaerobic digestion, this treatment system. So my research is mainly covers these four that topics along these treatment streams. And also not only this engineering or technology aspect, the science is very important. That's why I just use this photo as one example, because here in Ireland, you can see this seems everywhere, even within our campus. So the purpose of this one is just to make sure we can have that habitats for different types of that life. So that's something like how to achieve this biodiversity. That the concept of this one is the same as this microbial niche nexus. So we try to make sure in our system we can introduce more types of that microorganism, and also make sure they can function better. That they can have that diverse metabolic pathways, so finally can achieve our purpose. And also, as I just mentioned, for wastewater, it contains different types of that pollutants. So if we can enrich different types of that microorganisms, then we can remove these pollutants more efficiently. Because nowadays, we only know that only around 1% about these microorganisms. For others, we don't know what they are doing, so something like that. So for this, following these solutions, I just use this technology development as one example for our research. This is the post denitrification for nitrogen removal. This is something like that advanced nitrogen removal process. Usually, for advanced or tertiary that treatment, we need to consider about this footprint, because usually we just try to use that small volume that bioreactor So this should be quite important. That's why usually we use this biofilm based. For this biofilm, we just use this, different types of that carriers. For these carriers nowadays, usually we just use this, plastic, based, because the density of this one is quite low. That can achieve high filling ratio within this bioreactor. But another one is very important, for the size of this, carrier. If we have smaller size, usually the surface area should be higher. Then this can support more biomass growth. But at the same time, if we have that small size, that carrier, they can cause clogging of this, bioreactor. So we need to balance these two. That's why, for our research, we just, focus on this technology and develop different, types of that, reactor configurations, and also tested, all this on site. This is the first, that reactor I showed. For this reactor, we just use, ones that filling the reactor from the top, and then water flow down to the bottom. And also we can use that high filling ratio of this, plastic carrier. So, and also at the bottom, we use some, sand, so this can fill, this, maybe small that particles. But for other particles, it's mainly that, retained in the top, maybe in the top section. For this one, if we use that backwashing, this can remove, the particles efficiently. And also we examine, the performance and also the capacity, or maybe the microbial activity within this reactor. And this was developed in Kunming, and also further we received funding from Shenzhen, and then we introduced that intelligent that control system that uses that nitrate microsensor on site. So this can control the dosage of organic carbon. So finally, we can save the cost and also that improve the performance. And also we worked with, industry, different, companies on different types of this, technology. And this shows the performance of this sand-based, that post-denitrification that field systems. And also this was conducted in Kunming. And also in the lab, as I just mentioned, for this high ammonium that wastewater, we focused on these animals because the anaerobic ammonium oxidizing bacteria, because this bacteria is quite unique. So they grow very slow, but they have high capacity to remove that nitrogen. And also they don't require that organic carbon. So that's why for this one it's quite useful for high ammonium containing wastewater treatment. Because this one they can produce this red color. The pigment is quite special. So finally we can use different technology, or maybe use that biofilm, or maybe use that USB, this suspended that biomass to achieve the purpose. From all this technology aspect, we treat wastewater for the purpose of reuse. So that's why finally I contributed to that this ISO, that standard development. I was the convener, and also combined with other activities or outcomes from Professor Hu's that group. We received this award, because this award is quite unique, because this is the highest level in China. You can see here this the emblem. This only one type of the award that shows that emblem for this award. And also here it shows that my colleagues, this Professor Hu and this Professor Shuming Liu, now he's the Dean for School of Environment, Tsinghua University, and also this Qian Yuan, and he's the Dean Assistant in Shenzhen at the moment. The next, I will introduce about this knowledge generation. So as I just mentioned, for this engineering or technology, we need to understand why. So I think this slide I summarized a long time ago, but I'm very satisfied with this slide because here I just from the right to left, these are two typical reactor configurations. One is that plug flow, one is that completely mixed that reactor. So within these two types of that reactors, we can have that two types of that substrate gradient. If we have this completely mixed reactor, usually we have that low concentration of this substrate. If we have this plug flow, that will have that dynamic from the highest to lowest. So and these two conditions that can enrich two types of that microorganisms. One is that R, one is that K. Usually for R strategies, they can grow very quickly, but they can use that substrate more efficient. But for this K strategies, they grow very slowly, but they can use that substrate very efficiently. So they have high affinity for low concentration of this substrate. So that's why here we can connect ecology, that principles, to that engineering. And so goes back to my research. I'll just introduce or adopt this concept to our study. So usually we at the moment we are focusing on this anaerobic digestion. As you know, we can produce that methane from different types of that waste. Even for this one, that process for this one that includes different types of that microorganism. They work together to achieve this purpose. But for each step, there should be some limiting step, especially for this maybe here for methanogens and also that centrophs. They need to transfer or maybe break the limiting point. So that's why the electron transfer is very important for them. So that's why here I will introduce about this electron transfer between these two microorganisms. They can transfer electrons directly by contact to each other. So it's something like to produce that electricity. The electricity can be transferred directly. Another very good story, because when I can't remember the year, I visited this green gas, this company, because for this Senan, he did not attend the college or he did not learn any knowledge for this AD, but he, based on his routine work, he learned if the reactor cannot produce methane efficiently, he will dose more cow manure to that reactor. As we know, for the cows, they can produce that methane within their body, that's their stomach, because there are some methanogens there. So this is something like sometimes if we don't have knowledge, we can still operate the reactor or something like that. But for us, we know what's the possible reason. So anyway, this should be very good that experience. And also now goes back to our study. We introduced these two types of that reactor configurations, and finally we can enrich this electroactive that can produce that electricity, that bacteria, usually is geobacter. This can be enriched within this completely mixed reactor. So you can see the relative abundance is quite high within these two reactors. That should be completely mixed reactor. So that means now for this bacteria, this should be classified as that K strategist, because this understanding is very important for us. How to design the reactor, how to make sure they can be there and then they can be function. So based on this result, first we can enrich this type of that microorganism, and at the same time, if we use that ethanol, if you can see here, this is the completely mixed that reactor. this ethanol is mainly converted to that acetate. But if we use that, plug flow, this reactor, this ethanol can be converted first to that propanic, acid, and then, degraded to that acetate. So that means, the reactor configurations not only affect the microorganism, but also affect their metabolic pathways. So now, if we just summarize this one, that means this the basic engineered that system. They can have different levels of that scientific mechanisms. For example, at the metabolic pathways, that could be overflow mechanism or metabolism, within this plug flow reactor. From this community level, they can be classified as R and K, and also at this ecology that level, they can have different pathways. This could be something like the division of labor. And also based on these studies, I edited one book, and also especially I introduced all these ecological principles in each chapters. And also at the moment we are working on this topic by using that techniques from other disciplines, that including that mining, and also we are using that genome scale, that metabolic modeling, and try to answer these three questions. So this is the focus for my current study. And then following, I will introduce about this my ongoing activities, because for the previous that slide is mainly focused on that my contribution. Now not only research, I think that education is also very important. That's why now we just focusing on this marine career, this project that including 8 beneficiaries, and also we try to train that 14 that PhD students. And also some information is online, you can refer to here. For this project, and also designed this logo, and it shows this stressed condition, this wave that shaped line that indicate that stressed condition, and a different color that shows that different sustainable that development goals that refer to different aspects. And also we introduced DBTL, this cycle, because this one is very popular in that medicine or maybe other disciplines. And we try for different research tasks that can be classified to learn, and also how to design, and also how to test different ideas. And also for the training section, I will introduce this one especially about this table, because for this project we designed at least 3 training activities. One is that core knowledge, and then one is that techniques, and also that skills that should be trained for future that generations. And this is something like one module or one course. And also we designed this workshop for each year that based on also that design, and also beyond test, and also learn. So that's, I hope that in future this can be one module for our civil engineering, or maybe other that program. Finally, I just introduce about this future vision, because for my research, as I mentioned, for wastewater or maybe others, not only that high concentration of that pollutants, but also that low concentration of that pollutants, including that antibiotics, and also that other emerging compounds. Even their concentration is quite low, but their ecological risk or maybe other risk that usually that should be quite high. That's why we will focus on this low concentration pollutants removal, and we try to understand that microbial metabolisms from different levels, including that cellular or maybe community or ecology, and then we try to connect the water, not only wastewater, but also surface water, coast water, and we can achieve, for example, the technology or principle can be applied for bioremediation of that polluted surface water or maybe coastal water. And finally, we try to connect this water and also to energy at the moment, and also that health, maybe ecology, and finally, hope we can achieve this sustainability. For all this achievement, or maybe during my career, this wines that people, wines money. Maybe the idea is also very important, but anyway, here shows that my different groups from Shenzhen, now in Galway, and also that Jinan from Professor Xing, and also that on site in Kunming, we have lots of them colleagues there, and here I just listed the funding since 2021 because I was supported by this Galway University Foundation. This was quite helpful because during this period, I mainly focused on that research. That's also a big help, for me, because my English, was not that good. Because for the previous, 5 years, at least I can try to learn, and that should be a big, help. And also, this is the first funding I received because I think they are quite generous, for funding. Because for this funding, it was also quite helpful for first, that students, and also now I received, this Frontier Future, program from Research Ireland, and also some EU fundings, for this marine career, that actions. And finally, I think, from this talk, because I merely focused on that microorganism, they are quite small, but finally they can just solve, the problem, from that we are facing for that environmental problem. I think I believe, for each of us, if we can just, try to contribute, to each aspect, research or maybe education or maybe community policy, I think we can make, that impact finally. I think that's all, for my lecture. Okay, thank you. Yeah.
Great, thanks a million. And we have a minute or two for questions.
Yeah.
So first of all, congratulations. As I said at the start, it really is a significant achievement to be promoted to this level, and it's a recognition of all your achievements. And it's fantastic to be able to learn about the different aspects of your research and the focus of it. I'll give people an opportunity to ask questions, but I might ask one first. Just in terms of scale of implementing this now within the Irish system as a kind of a wastewater refinement, how, in terms of scale of experiments that are done here in this building versus what would be needed to start to implement it maybe for restoration of ecological or addressing problems or for energy generation, what's the scale up for those kind of initiatives?
I think for in Ireland, because of the government planned, to build more than 300 that AD plants in future, so that should be big market for the next at least 5 to 10 years. But to be honest, for myself, at the moment we are just focusing on that basic or maybe fundamental research, because I don't have too many connections with industry. I hopefully that in future, if Owen or maybe from the school or maybe college, if we can have some more connections, then we will consider about the actual application. But the principle.
Absolutely.
That can be applied, definitely, for actual plant.
It's great, clear from your publications, and that these are fundamental scientific questions that need to be solved, understood. There's a lot of discovery in it.
Yeah.
And then obviously beyond that is first and foremost the fundamentals need to be understood. So great.
Yeah.
Does anyone else have a question there now? Yeah.
Well, not so much a question. I just make a comment that I did my PhD at the same time as Guangxue, and I learned an awful lot from him. And I'm not surprised to see how successful you've been. And congratulations. And also,
thank you. Yeah.
I still learn a lot from you because every time I'm an internal examiner on one of your students' PhDs, it's a continuous learning process for me. So it's very interesting, and thank you very much, and well done on your talk today.
Yeah. Thank you. Yeah.
Well, we'll have opportunity afterwards to, there'll be a bit of food and people might want to talk and ask questions further there, but and keeping an eye on time, we're bang on time now, so thanks for keeping to time, and we'll follow up afterwards. But again, we just give you a big round of applause before we move on to our
Thank you. Okay, thank you.